Hi, guys. Hi, Felicitas.
Hi, Lee. Thank you so much for joining on time. We're just
Yeah,
waiting. We're just waiting to get for the time to get 1:30 and then we'll start. And thank you for everyone who's
yes.
joined so far. Thank you.
Also, Tor Vaughan is going to co-present with Christine and I. So when he joins, can you also make him a panelist?
Perfect.
Thanks. Hi, Christine.
Hey, Lee.
Do you want to practice sharing your screen?
Yeah.
Are you also wearing black? Should I go change? I have purple earrings at least.
Yeah.
Okay, let's see. Oh, no, she's sharing her screen, but you should be able to, right?
Yeah. I'm gonna just try.
So, hello, everyone. We'll just give people 5 more minutes and then we'll start at exactly 1:35 p.m. Thank you. Good afternoon, everyone. Thank you so much for joining us. We'll start at exactly 1:35 p.m. Thank you. So far, we have 34 participants and we have all our speakers and presenters with us. So we'll start at 1:35 p.m. Thank you. For those who are joining us, thank you. We'll start at exactly 1:35 p.m. So it's exactly 1:35 p.m. and good morning, good afternoon, good evening, depending on where you're from, where you are. Welcome to this webinar. We are so delighted to have you join us today. Our webinar today will be around advancements in soil and land health monitoring for ecosystem restoration, biodiversity, food and nutrition security, and climate action. It is hosted by the African Population and Health Research Center in collaboration with CIFOR-ICRAF. And maybe just to mention briefly, APHRC is a research to policy institution generating evidence, strengthening research and related capacity in African research and development ecosystem, and engaging policy to inform action on health and development. We all know more about ICRAF. When you hear ICRAF, I know we all think about trees, but to just add more around ICRAF, ICRAF generates and applies evidence to meet today's most pressing challenges, specifically around energy insecurity and the climate and biodiversity crisis. So thank you all for joining us. Today's session, we are gladly to be joined by Dr. Elizabeth Kimani Morage, who's the principal investigator for the project itself. The webinar itself is under the Visibilize for Climate Action Project, which is a project funded by Wellcome Trust. Dr. Liz will speak more around the project itself once we start, and then we're also glad to be joined by Christine, who works for ICRAF, and also Dr. Lee, who works for ICRAF. I'll share with you in a minute the bio of the speaker, and then we'll go later, and then I'll share later on the bios of the presenters. Thank you. Just give me 1 minute before Dr. Liz comes in to give the opening remarks. I can just share a brief bio of who she is and the work that she's doing. And kindly, you can also engage with us online. Hashtag is Visibilize for Climate Action, tag at APHRC, and also tag at CIFOR-ICRAF. So this is Dr. Elizabeth Kimani. She is she's the public health nutrition specialist and a senior research scientist, and she leads the nutrition and food systems units and the health and well-being theme unit at APHRC. She's also the principal investigator for the Visibilize for Climate Action project. She's a dynamic and impact-oriented African research scientist and a leader. She transforms lives in Africa and promotes human rights through research, public engagement, policy influence, and capacity building. She champions research on maternal and child nutrition and zero hunger in Africa. Her current work focuses on food systems transformation and the interlinkages between climate change, food systems, nutrition, and health. And she's leading the Visibilize for Climate Action Project, which is a Wellcome-funded project on the interlinkages between climate change and health in Eastern Africa to catalyze policy and practice action at scale. She holds a PhD in public health, focusing on the double burden of malnutrition in South Africa from the University of Witwatersrand, South Africa, and a Master's of Public Health degree and a bachelor's degree in environmental health, both from Moi University, Kenya. She's also an advocate of the High Court of Kenya with a special interest in human rights law, holding a degree from the University of Nairobi and a postgraduate diploma in law from the Kenya School of Law. So thank you so much. I'll now hand over to Dr. Elizabeth Kimani, who will share with us the project overview of the Visibilize for Climate Action project, and she'll also welcome us to the webinar. Thank you. Over to you, Dr. Elizabeth.
Yeah, thank you. Thank you so much, Feli, for that kind introduction. So welcome everyone to this webinar. I'm so delighted that we have this webinar today, and we are going to be hearing a lot about how climate change impacts on soil health, ecosystems and soil health included. And I think at this point, we all know that climate change is having a lot of impact on ecosystems, on human health. And that's why us at APHRC with our partners, we are very interested in this focus, and we are working to really make visible the climate impacts on health and how that happens through different pathways. And that's the focus of this project called Visibilize for Climate Action. So we want to really make visible the climate impacts on health to catalyze action at scale. And so we are doing a lot of engagement, and this is one of those engagements that we want people to be able to understand these interlinkages and really be able to see how we can all work together as a team to be able to come up with solutions. So this afternoon, we are delighted to have Dr. Lee and the team from ICRAF presenting to us about how climate change, the interlinkages between climate change and land health and soil health, and how the impacts. So I'm going to present about the Visibilize for Climate Action to start with, and then we'll have the team from ICRAF present about soil health and land health. And I invite all of you to join us and really be actively involved, ask questions, and also contribute your perspectives on this issue. So let us have a warm discussion about this topic. Thank you. So let me just project my slides to present about Visibilize for Climate Action project. Kelly, I hope you can see my slides.
Yes, Dr.
Yes.
Elizabeth. Yeah.
Okay, good. So this is the topic of the day, which will be elaborated more by Lee and Tim in their presentation. So, as I mentioned, that climate change is really having a lot of impact on the land and soil and the ecosystems, and this leads to poor health, human health. It's often said that health is the face of climate change, and so for us to be able to protect health, we need to really to come in to understand these interlinkages, how climate change actually impacts on health through different pathways, including the land degradation and soil health, and that's why we are talking about this today. So this has a lot of impact on issues of nutrition and food security, and also consequently mental health, and the mental health burden is rising in the Eastern African region, globally, but also in the Eastern African region. And the problem we have is that we have evidence gaps. We have limited local evidence that based on local research, And then we also have a gap in policy uptake. And that's why it's important that we do this kind of work locally so that we can inform context-specific actions. So, I will talk briefly about the CBLIs. I'll just introduce the project and what we are doing so that we understand the focus. And this is, it's focused on visibilizing climate change impacts on nutrition and mental health among vulnerable populations in Eastern African drylands to catalyze climate action at scale. So it's a 3 or so year project funded by the Wellcome, and it's led by the African Population and Health Research Center, which is APHRC, working with other partners, and the other partners are indicated with their logos here. We have ICRAF, and we have other organizations that you can see on the slide. So we aim to make visible through research, public and policy engagement, the impact of climate change on nutritional status and mental health of vulnerable populations in the Eastern African region to be able to catalyze action at scale. So this is the focus of the project. We have a research focus, we have a public engagement focus, and also a policy engagement focus. So under the research, we want to be able to quantify climate impacts on nutritional status through attribution science. And when I talk about nutrition, it's not just the nutritional status as relates to health, but we are also talking about crop production or food production. So the impacts of climate change on food production and food security that relates then to nutrition. We also want to be able to document lived experiences with ecological, food security, nutrition, and mental health impacts of climate change. So how people have experienced these impacts in their communities, in the different communities. We are also doing landscape surveys, and this is where we have soil and land health that we'll be talking about today. And we're also assessing readiness for practice and policy change. And we are also modeling future health and ecological impacts and estimating the cost. So we are not just collecting primary data or working with secondary data, but we are also modeling to see the future scenarios. And we're also doing a case study of agroecology as a climate solution. So this is integrated in the research work that we are doing. For public engagement, we want to enhance the understanding on the interlinkages between climate change, food systems, nutrition, and health, and also communicating evidence. So this is at the community level, but also general public, using different methods, engaging communities at the community level, using participatory methodologies, but also working with media and other ways of communication to be able to communicate widely in the public. We are also doing policy engagement to promote evidence-informed decision-making for policy and practice. We have been doing policy analysis of policies, like climate policies at the national level, but also at county level in Kenya, to be able to understand the integration of health core benefits in these policies, and also finding opportunities to catalyze inclusion of health in these policies. We have also been doing policy engagement, and we continue doing that, and policy advocacy. So the study is, we are doing this work in Eastern African drylands, Kenya, Uganda, and Tanzania, and Ethiopia, with a deep dive in Kenya. And so the primary work, the primary research and engagement are happening in Kenya. We are doing this work in Turkana, which is an arid area, Samburu, semi-arid, and Laikipia, which is dry, subhumid, or the areas we are focusing on are those agroecological zones. So what makes this project unique? This initiative is locally led, but we have international collaboration, and it combines attribution. The research itself combines attribution science, modeling, and also lived experiences. It strongly includes community experiences as sources of evidence, because it's very important, because the lived experiences are very rich sources of data. It also actively combines research, public engagement, and policy engagement for greater impact. So these are going side by side. We are not just prioritizing one over the other. So it's not just a research project, but really an impact-oriented project. So it's action-oriented, involving policy and practice, to inform policy and practice change. Yeah, thank you so much. So now I return it to Feli to tell us what next. Thanks.
Yeah, thank you so much, Liz, for that overview about the project. And just to also emphasize, the webinar today is a key activity of the project work stream on soil health and ecosystem restoration, and contributing to the broader mission of linking evidence to policy action, and also practice and public discourse. So the next step of the webinar will be to have ICRAF share their findings, specifically around advancements of soil health. Around the webinar today, they'll be our key presenters. And before they start the presentation, I'll just give you an overview of who they are, on who Lee is, and also who Kristen is. So just give me a minute. And also in the meantime, if you have any questions, please feel free to utilize the Q&A section. Thank you. So we have Dr. Lee Ann, who is the Global Research Lead for Soil and Land Health at CIFOR-ICRAF, based in Nairobi, Kenya. She's a soil scientist. Her research focuses on landscape restoration, understanding drivers of degradation, and quantifying the impacts of land management on soil health. Since 2009, she has co-developed and implemented the land degradation surveillance framework in over 45 countries across tropics. This framework is a systematic methodology to assess soil health, land degradation, and vegetation dynamics, and track restoration efforts across landscapes over time. She has published widely on soil organic carbon, ecosystem services, and land degradation across sub-Saharan Africa and the tropics, including a coherent set of open access data sets. She co-founded the Coalition of Action for Soil Health, which aims to catalyze investments in soil health for human well-being and environment. She's on the scientific task force and the monitoring task force for the UN Decade on Ecosystem Restoration, a member of the expert network of Regen 10, and she's a founding board member of the International Union of Agroforestry and the board of directors of the International Fertilizer Development Center and VADA Foundation. We also have Christine, who also is a research associate at ICRAF, and she works on soil and land health research and engagement, leading work at CIFOR-ICRAF, where she helps ensure soil and land health research data and evidence is accessible and useful and makes impact. She has over 7 years experience working on soil and land health research, ecosystem health monitoring, and stakeholder engagement with the aim of enhancing ecosystem resilience. Christine has experience in leading farmer-centered restoration activities, large-scale data collection efforts in the field, data extensive experience leading stakeholder engagement to enhance the use of soil and land health data and evidence. So today we are very privileged to have two experts to share with us more information around the work on soil, not just soil, but generally healthy soils. So over to you, Dr. Lee or Christine, either of you can take lead on this. Thank you.
Thanks, Felicitas. And we will start with Christine Magaju. She will start the presentation and I'll talk until she shares her screen. We'll also be joined by Dr. Tor Vaughan, who will present their predictive modeling. So thank you so much for this opportunity. Over to you, Christine.
Well, let me know once you see the screen, Lee.
I see it.
And it is in presentation mode.
You need to swap displays because I
okay.
see the other display.
All right, let me just do that. Swap displays. Wonderful.
And today we're very excited to talk about the advancements in soil and land health monitoring for ecosystem restoration, but also looking at the comments in the chat. I would really like people to be thinking about how we can look at this to understand the impacts of climate change, how we can use soil health data to understand the nutrition of our crops, of the food we eat, and of the animals and of ourselves. So please put lots of questions in the chat, and I'll hand it over to you, Christine. Thank you so much.
Great. Thanks, Lee. And thanks everyone for joining the webinar this afternoon. It's really exciting to be able to share some of the results and some of the work that we're doing around soil and land health monitoring. But just pivoting on what Dr. Liz and Dr. Lee talked about, like the global importance of soil, right? We grow our food from soil, but we're not managing them properly. With as little as basic soil management, we can produce up to more than 58% more food. So echoing back to the need for nutrition, all of these things that we just heard about. However, we're losing tons of soil every year due to degradation. 35% of the earth is degraded, and soil erosion is a most widespread form of degradation, impacting about 3.2 billion people. And in Kenya, we're losing over 20 tons per hectare of fertile soil every year to erosion. So we need to shape up. We need to make investments into restoration and managing ourselves. So what do we mean when we talk about healthy soil, right? We need healthy soil, but what does that mean? Healthy soil is the ability of the soil to sustain the productivity, the diversity, the environmental services of terrestrial ecosystem services. And this is from the Intergovernmental Panel on Soils, emphasizing on everything we heard about the centrality of soil when it comes to our very own existence. And going into, why are we doing this, right? Why are we monitoring? And the broad objective is to provide land and soil health information in Laikipia landscape, as well across all the other ecosystems, and we'll talk about it in a minute. Specifically, it's implementing the land degradation surveillance framework, and I'll tell you more about it in the next couple of slides, in the Laikipia landscape and elsewhere, but also assessing soil health, land degradation, and vegetation diversity. But it's also about linking land and soil health with farm and rangeland diversity, communities with resilience, and nutrition, and dietary diversity. So bringing in the aspect of nutrition as well. So before we jump right into the monitoring, I do have a question for you, and I'd like to invite you in the chat to indicate if you've conducted biophysical sampling before, and how have you done it, but how have you used this data, the resulting data from the biophysical monitoring. So take 1 minute or 2 minutes, just write in the chat if you've conducted biophysical sampling, the data that you got from the sampling, and how you use that data. And I'll ask Lee to help me check with in the chat just to see what is what we're saying in terms of our experience. So let's take a minute to do that, and then we'll continue.
Great. And the reason we ask this is also because it's really important that we break silos, right? We'd love to have some social scientists also think about soil and land, climate scientists. So it's just really interesting to see, who has been integrating and collecting this data. And I do notice, Felicitas, that we can only send messages to hosts and panelists. I'm not sure if it's possible to send messages to everyone, because that may be a bit difficult. So maybe as we're thinking of that question, and we do have a Menti poll later, so we can just keep going, Christine. Thanks.
Wonderful. Thank you. So for the next couple of slides, we'll talk about monitoring, right? How do we monitor soil and land health? And at CIFOR-ICRAF, we use the land degradation surveillance framework. And this is a comprehensive method for systematically assessing soil and land health across multiple scales. It provides a method, as mentioned, for assessing soil and land health, and provides a consistent set of indicators for monitoring ecosystem health, including vegetation cover, structures, vegetation cover and structure, looking at trees and shrubs and grass species diversity, as well as land use. So before I tell you and go into a deep dive of the indicators monitored using the LTSF, I'd like to take a minute to go on Menti to see what are we monitoring, what are we using, what indicators are we using to assess soil health in our projects, in our organizations, but also which ones we actually monitor. So you can, if you're on your phone or if you have your phone, you can scan the QR code or go to menti.com and enter the code 2496-8332 and then answer the 2 questions. So what are the indicators that we can use for assessing soil health, and which ones do we actually monitor? And please do let us know if you're having trouble connecting the Menti. But yeah, scan the QR code or go to menti.com and enter the code below.
Leah has also shared the link on the chat, so you guys can also have access to it here.
Great.
Yep, I've done that. Great.
And I see, we're monitoring or we're using soil erosion, monitoring soil organic carbon, soil microbes, looking at soil biological activity, looking at productivity, microorganisms, looking at microorganisms, soil texture, soil structure. Great. Nitrogen, salinity, and erosion. All right, I don't see any other, but let's add to the chat, and carbon. So please add to the Menti poll in terms of what some of the indicators for assessing, but also the ones you actually monitor. Great. All right, so what do we use for the land under the land? Sorry, let me just close that. In the land degradation surveillance framework, we monitor multiple indicators in the same georeference location. And you can see on the right, it gives you a list of all the indicators monitored. So includes indicators of land use, land degradation, impact on habitat, land cover, and also indicators of soil health, some of what has been already listed, looking at soil organic carbon, total nitrogen, infiltration capacity, texture, as well as the microbial activity. But also one of the key aspects of the LTSF is the systematical, it uses a hierarchical sampling design. And at the top of the screen, I don't know if you can see my cursor, it tells you, so it shows how the sampling design is structured. So you have an LTSF site, and that's it can be randomized on interventions, on areas of interest, and then within each site you have clusters where sampling takes place, and within each cluster plots. So this robust monitoring allows us to reduce bias, but also to be able to do robust monitoring. And on the left of the screen is the LTSF manual, which you can download on the link below. Great. So the result of systematically monitoring same indicators across georeference locations means that we're able to develop a data-driven network that assesses soil health, land degradation, and biodiversity. And this, the map on the screen shows you where the LTSF has been implemented in over 45 countries across the global tropics, with a keen interest in Africa. And the green dots are, the sampling plots from which where the data has been collected from. So this has enabled, as I mentioned, robust monitoring and statistical comparisons across different land uses, including rangelands, right? For the rangelands within the LTSF, the module assessments are done every 2 meters on a 30-meter transect. That's 2 30-meter transects, so northern, south, and east and west. And at each 2-meter point, we assess the indicators on the screen. So the nearest perennial grass, the nearest annual species, looking at bare ground, looking at if there's princes of dung on the point, if there's leaf litter, but also the woody species and the nearest bulb species. So for each point, looking, as I mentioned, what is the name of this, the perennial grass species, the name of the annual grass species, and the FOBs. But also beyond that, it's about the engagement around the data and the monitoring. So we, bringing the community, stakeholders, and everyone along to collect the data, but also to share back the data with them and reflect and interrogate those data sets. So what makes the LGSF very unique? You're able to monitor across a range of landscapes using very low, very easily accessible tools and equipment. And on the left is a screenshot of literally all the tools that you need to be able to conduct an LGSF, to do an assessment using the LGSF. And a lot of these are fabricated locally. But beyond that is the hierarchical sampling design that I talked about it. But all the field data, all this data is enabled in advancements in soil spectroscopy, but also machine learning and remote sensing. And we'll hear a lot more when we go to the results. And as I mentioned, so this is just to highlight some of the activities within the data collection for the LGSF. So you have soil sampling, you have collecting soil data using the augers, and we collect data from both topsoil and subsoil. So 0 to 20 centimeters and 30 to 50 centimeters to see how the different indicators vary across depth. Right, and this is just another pictorial of the data collection process of collecting the data, but also, as I mentioned, doing vegetation transects, being able to understand both vegetation density and diversity across rangelands, but also other ecosystems. Being able to do tree measurements, for biomass, but also to be able to understand vegetation dynamics from density, diversity as well. And doing infiltration capacity, right? Understanding how fast the soil water goes into the soil, which is a good indicator for when it rains, when the soil is saturated, and how fast does the soil get eroded. Being able also to understand the impact of some of the land management, agricultural practices on some of these land health dynamics. And all of that is underpinned with electronic data entry, allowing us for robust checks to be able to assess and analyze the data as it comes along, but also and training the stakeholders as well to be able to collect such data. Right, I think at this point I'll hand over to Lee to take us through implementing the LGSF in the Visualize project and share some of the results. Over to you, Lee.
Sure. And maybe before we start, sorry about my video, does anyone have any questions on the methodology? Because we really just don't want to be talking at people. So if it's possible to take some questions on the methodology, and then we'll talk about how we've implemented in the Visualize for Climate Action context. So Felicitas, are people able to raise their hands, or how is it possible for people to engage?
Yes, they could raise their hands, and they could also utilize the Q&A section. So they are free to raise their hands. We'll allow them to speak. So if you have any question, please raise your hand. We'll allow you to speak. And if you are not comfortable speaking, you can also just write your question in the Q&A section.
Great. And this is really interesting. So MKEWP has trainer of trainers. And I think that well, to everyone, we extend an invitation to the CIFOR-ICRAF, to the ICRAF campus in Gigiri to tour the soil and land health, the tree diversity, and the spatial lab, because I think there's a lot of synergies here. And it's nice to meet you, Dr. Janeth. So soil carbon, this is absolutely fantastic. Great. So thanks, Christine, for changing the slides. I'm going to talk about implementing the LGSF for Visualize for Climate Action. Here's a photo of some team members in the field. And as you can see, as Christine indicated, we sample in a 10-kilometer by a 10-kilometer grid. We now can modify our sampling to do a more stratified zone, but for this project we did, if you go to the next slide, a 10 by 10 grid. So you can see here, there's 16 clusters of points, and those are all the sampling points. So you have in each one of these clusters, 10 plots. It's just west of Rumaruti town in Laikipia West. So those are the 160 LDSF sampling plots on a 10-kilometer by 10-kilometer grid. On the next slide, we will show you some things about the soil sampling. Next. So as Christine said, we took soil samples using an auger at top 0 to 20 and sub 20 to 50 centimeters. And yes, I said there was 160 plots. One of the plots was not accessible, so we have 159 topsoil samples and 156 subsoil samples. Now, why do we have less subsoil samples? When you have compacted soil, sometimes it's very hard to get that subsoil sample out of the ground. So in that case, you would record a restriction. So actually 3 of the plots could not get to the 50 centimeters, indicating restriction or compaction. So the next graph is really about some of these data. So all of these samples were taken to the Nairobi lab. And like I said, everyone has a standing invitation to come visit us. I'll put my email in the chat. We love hosting people and providing tours. This is Dominic drying the soil samples. Then we analyze them in our own lab here in Nairobi at the APHRC campus. So now we're going to get to some results. So soil sample results. And I'm really glad that Christine asked about what indicators, because I think someone said pH. Now, I'm going to just take a minute to describe this graph, because right now it kind of looks like a drop of water, or maybe it looks like a squiggly line. Not really sure what everyone's thinking. But in fact, this is a graph that in the blue box plot shows the variation in soil carbon in topsoil and in subsoil on the right. So the box plot is showing almost the same thing as the density plot on the left, except it just is a different visual. So basically you can see that the average pH was about 6.2, or the median pH was topsoil was about 6.2, where that black line is, but you had some plots that all were above 7.5 and some that went below 6. So why do we sample so many plots? It's because we want to understand the variability. Because if we make recommendations on just taking one sample for the whole landscape, the farmer with the pH 7.5 is going to have a very different management practice than a farmer who has a 5.5 pH. On the right is the same, is the subsoil pH, and you can see, oh, sorry, keep going on the pH. So we have the topsoil and the subsoil, and basically a similar pattern, except there were subsoil samples that had even higher pH, above 8. So this is very interesting when we talk about crop suitability. This is very interesting when we talk about management techniques, composting, application of ash. So the next slide, thank you so much, Christine, is around clay. Does everyone know what clay is? When you grab a piece of soil and you squeeze it, you can tell if it's very sandy or very clayey. In this case, Rumaruti soils were very high clay. They had over 50% clay in the topsoil and the subsoil. Again, you have the box plot, and then you have the sideways density plot. Does anyone notice which had higher clay content on average, the topsoil or the subsoil? Can anyone see the graph? Which was higher? Is the median topsoil or the median subsoil higher? Anyone? You can write in the chat. It is the subsoil had higher carbon. Yes, thank you, Eugene. Good. And I'm just responding, yes, the APHRC campus in Nairobi. So this is a common phenomenon where you have leaching. Thank you, Jenith. So where you have, thank you, Eriku. This is great. So you can often have higher clay thanks, Consolata, nice to see you. Clay content as you increase with depth. So this is also very interesting. So the next graph, and I promise we only have one more of these, but now we'll get used to seeing box plots and density plots. Whoa, now we see a very different pattern here, right? So this is soil organic carbon in grams per kilogram. So thank you, Joseph. I love this. So Why do we look at soil organic carbon? We look at soil organic carbon because it influences so many processes and we can improve it with management. So if we have higher soil organic carbon, we can have higher water holding capacity. We can have better stability. We can have increased nutrient availability. So we like to say soil organic carbon is a key indicator of soil health. Now, here's our box plot and the sideways density plot. And here, remember, these soils have about 15 grams per kilo in the topsoil, which is about 1.5%. This is actually about where we would hope it to be. What we don't want too many plots of is below 10, below 1%. And you can see there's about 10 plots that fall in that below 1%. So what we would like to do in this project is use these data to target and say, let's really try to work on these farms to make sure we maintain the good soil carbon content, but that we increase the soil content with manure application, with agroforestry, with soil water conservation. Now, here, I don't know if you guys can hear that. That's my dog's tail beating the floor. He must be very excited about me talking about soil carbon. This has a different pattern, which has, thank you, which has a lower carbon is the subsoil. So you can see the subsoil average is around 10, maybe 11 grams per kilo, where the topsoil is 15 grams. So in this situation, and we really want to have interventions that reduce erosion and also increase soil carbon. This would be very important for the productivity and for nutrient availability. Is there one more slide, Christine? Great. Done with the soil. In LDSF, thanks, Akuro. Yes, I think he's getting very excited. So now we're going to talk about the vegetation surveys. So on the next slide, we're just going to go through and highlight the different species that we observed in the plots. So remember, the LDSF plot is 1,000 meters squared. So I'm going to go to my slide because I don't see it changing. And basically, we looked at all the trees and shrubs in each of the subplots and their herbaceous cover rating. Now, one thing that we observed is that we had many, many plots, almost 70 plots that had, this is very good, sorry, herbaceous cover of over 65%. So that means that you can see this little schematic here, that this means that the ground was covered. Very few plots had low herbaceous cover. So this is incredibly promising. And what we would want to do is these plots in the middle, the 10 to 14% and the 40 to 65%, we would want to encourage those farmers to increase their herbaceous cover. What does this mean? We say soil is like the skin of our earth. When we expose bare skin to the hot sun, it's the same as exposing the bare soil to the hot sun. We want to keep it covered. We want to reduce the wind, the temperatures. And so we want to hold on to that moisture. So most of the plots, 70 of 159, had good herbaceous cover rating, but I think we need to work with these other farmers to make sure the soil is covered. And thank you for the question about pH. Why was it lower in the upper layer? Yes, so that's often because where you have vegetation, composting, it actually can increase or lower the pH. So that is often why you will have higher pH in the subsoil. Evans, please, if you can unmute yourself, come on in. I see you have a hand raised, Evans Omondi. Can you unmute? Okay, well, Evans figures out how to unmute. We will keep going. So the next slide now talks about the woody cover. So the woody cover shows a different pattern. And in this landscape, there is lower woody cover. So you see the highest graph, the highest number of plots on the right was less than 4% woody cover. And this is also because we have rangelands, right? So we don't expect we're not in a forest. We don't expect 65% woody cover, but it is interesting to see the distribution of plots. So we had 50 plots with less than 4% woody cover, 25 plots with 4 to 15% woody cover, 18 plots with no woody cover. So this is the categories, and we had 0 plots with greater than 65 woody cover. Next slide basically shows species. So we're very excited about this. So in the LDSF, a tree is something that's woody vegetation that's greater than 3 meters tall. So can anyone read on the graph which was the most common tree species in the Rumaruti LDSF site? Who can write in the chat? So this is a species occurrence graph. So the longer the bar, the more common the species. So we only had 8 species of trees. Who can name what was the #1 most common species? Eucalyptus saligna. The next one was cypress. Then, yes, eucalyptus. Exactly. So you have to get down to the 4th most common tree, which is Acacia sayal, which we know grows in waterlogged areas and is photographed here. So if there were some interventions here, we would really need to work with the farmers to understand why they're growing so much eucalyptus in the drylands and see how we could nudge to increase the occurrence of indigenous tree species. So when we're thinking about interventions, Liz, Felicitas, these are the kind of data we can use to say, okay, what are the indigenous species that we can also work with? So the next graph is shrubs. So we could also combine these data, but we keep them separate so that we have a structural understanding of the tree and shrub across the landscape. Now, we actually have 15 shrub species. So a shrub is 1.5 meters to 3 meters tall, woody vegetation. In this case, most of the shrubs were indigenous. So we have Euclea divinorum, which is photographed, Croton dichogames, which is also photographed, as the top shrub species. So it is positive that we have indigenous tree species here. Yeah, it's a very good question, Simon. I mean, I think they're growing cypress and eucalyptus for timber. They're using these trees. So now it's really about how to nudge to also include some of the indigenous species there. So I hope you enjoyed thinking about soil, at least as much as my dog did, and thinking about vegetation diversity. So now, how can we use these data to do predictive mapping? And we're really excited that Tor Vaughan, we'll have to Photoshop his face into the flyer. He's here to really talk about this. And I just want to check again if Evans, if your hand is still raised, if you wanted to say something, I don't know if you can unmute yourself. But it looks like your hand's raised, so I wanted to give you the opportunity to come in. But if you can't come in, I understand it's a Zoom thing. So I hand it over to Tor, and thank you again to Christine for changing the slides. Thanks.
Yeah, thanks, Lee. So I'm going to just cover a little bit about in terms of how we scale the types of assessments that Lee and Christine were talking about. So essentially in the LDSF, as you saw, we measure things at the plot level. So we have these plots across landscapes where we do soil sample collection and assess the vegetation, etc. So we also then need to look at how can we scale this across larger landscapes? How can we use it together with other types of data sets, including what Liz was talking about in terms of mental health, in terms of nutrition, in terms of household survey data sets, etc. So for this, we need to leverage a number of new technologies. And these are areas that are moving very fast, and this is a very fascinating space at the moment. Years ago, we started at APHRC working on soil spectroscopy, which is illustrated on the left there with some of the guys in the lab and some of the samples there, where we basically just shine infrared light on soil samples and then predict soil properties using these spectral signature or fingerprints of each soil sample. So basically that allows us to do soil analysis much faster, but also much cheaper, cutting costs by about 90% relative to to conventional methods. So that's one aspect of scaling that's really important. The other part is using remote sensing or satellite images, but also other types of remote sensing technologies to map various aspects of soil and land health across these landscapes. So the middle there shows an example from Laikipia of fractional cover developed by one of our team members, Nathaniel Robinson, kind of combining things like tree cover, shrub cover, grasses, and bare ground. But we can also do this for different types of soil, for soil properties and other types of indicators. So if we go left, next, sorry, Christine. So basically, this is the approach that we've been taking it within Visibilize as well, mapping these indicators. And you saw Lee's graphs on pH and carbon. So here are two maps showing soil pH for 2023 and soil organic carbon on the right for the 3 counties that are part of the project. So you can see here for Laikipia, for example, and across to Turkana, there's huge amounts of variation in terms of both of these soil properties. In Turkana, we have areas that are shown here with in very dark purple. Those are areas where we have high pH, so higher than 8, 8.5 often as well. So those are areas where we expect big, important constraints in terms of salinity and other things. And this is really important to understand and be able to map in order to inform management. These areas are also some of the same areas where we see low carbon on the right-hand side. So the lower carbon values are lighter in colors, where the higher carbon values are darker or brown in color. So we can basically then map these properties across the counties in a continuous way, which allows us to then use this information in combination with other types of indicators. Next, please. So the important thing here is really understanding dynamics, right? Both in space and also in time. And we're not just limiting this to soil properties, of course. So we can look at things like tree cover on the left here. Again, a lot of the areas where we saw very high pH, low carbon, we also see low tree cover within the Turkana area here. We can see Mukogoro Forest, for example, in Laikipia. We can see the Matthews Range in Samburu, and some of these other mountain ranges where we have higher tree cover within these ecosystems. And on the right here is a very important indicator, and that is soil erosion prevalence. So again, huge variations. We can see like parts of Laikipia with relatively low erosion prevalence, but also pockets with very high erosion. And again, in Samburu, some of the forested systems with low erosion in blue and higher erosion in other parts. And then again, when you go to Turkana, the same areas where we had low pH, high pH, sorry, low soil organic carbon, we also tend to see high erosion prevalence. So, and this is not because the models that we use know that there's low carbon and high pH in these systems, but they happen to sort of coincide in a way. So these models are independent of each other, but they can be used, the results can be used together to really give us a comprehensive assessment of soil and land health across these landscapes. I'm not sure what's next. Go ahead, Christine. Yeah, so one part of this that is we've been focusing a lot on in recent years is, and then one thing is to sort of use these very scientific and robust approaches that we've talked about so far. And this is all really important, of course, because we need to build these data sets and we need to build the evidence base around this to give us a level of rigor that allows us to inform interventions and inform decision-making at various scales. But also it's really important to look at how we can involve communities in restoration and in monitoring, not just in rangelands, but across ecosystems in general. So we developed a few years ago the Regreening app, which we have rolled out across a number of different countries, and it has over 300,000 farmers that are using it at the moment. And it's an app to do citizen science data collection on a number of different things, including farmer-managed natural regeneration, tree planting, nursery establishment, and more recently also rangeland restoration. Next, please. There's a QR code there. If you want to scan that, it's available on the Play Store for free. So just to kind of summarize some of what we've been discussing so far, what we're working on is basically combining the systematic field sampling using the LDSF with remote sensing, and also the use of spectroscopy, so new technologies, as I just talked about, and then using that in conjunction with assisted citizen science to geo-reference interventions to understand what farmers or pastoralists or organizations and projects are doing on the ground. So the combination of these things is a very powerful way to understand what types of interventions that are going into landscapes and what the impacts of those interventions are. And with all of these combined, we can also develop really interesting ways of, visualizing and interfacing this with users through interactive dashboards, and so on. And this is something that we use extensively in our projects to inform decision-making at various scales, also at the policy level, but for the most part, kind of more at the project level. So giving users the ability to interact with these types of indicator maps, the remote sensing, etc., to inform their decision-making.
Great, thanks, Tor. Right. Thank you so much, Tor and Lee, for the discussion and the questions. So I'll open it up to the rest of the participants as well. Like there are already some questions into the Q&A, but please feel free to unmute. Give your reflection, give your feedback, but also ask your questions. We can start with the ones already in the chat, in the Q&A, and I'll some more answered. But there's a new question from an anonymous attendee who will ask if the higher organic carbon is associated with reduced level and shifting micronutrients in the edible components of the plants growing on these soils. Lee, do you want to take that one? And there's also a question on monitoring, Tor, that asks, from Dennis, that asks, do we have soil health monitoring kits for on use, for farm use that are kind of a citizen-led science approach?
Great questions. Excellent. So yeah, I see if it's higher. So yes, higher organic carbon in the topsoil is associated with the inputs, right? So leaf litter, composting, reduced erosion. So this is absolutely how we can increase soil organic carbon. It takes longer to get to the subsoil, but thank goodness for termites and earthworms that can the bioturbulence in the soil that can bring carbon lower down. I also saw a question about how do we overcome challenge of very rocky areas? Absolutely. So in rocky areas, it's still important to sample there and understand what the vegetation diversity is. You may not be able to get a soil sample there, but you can record it so that we understand the variation in the landscape from the rocky areas to the deep soils. And thank you for asking for the LDSF manual. I'm going to put it in the chat, our LDSF website. And I think it's over to Tor. Maybe you could repeat the question again. Thanks, Christine.
Thanks, Lee. Yeah, the question for Tor is, do we have soil health monitoring kits for on-farm use that can have a citizen-led science approach?
Yeah, so in terms of kits, the answer is not yet. I mean, there are kits out there, but we've done a number of assessments of some of these, and I wouldn't say that we are at a level yet where we would recommend these in terms of accuracy, because we do want to make sure that the information that we're giving to farmers is accurate enough for them to make meaningful decisions. But one thing that I can say is that we've been making a lot of progress in terms of the predictive modeling that I talked about. So we are working on ways to provide recommendations to farmers based on predictions that we make from remote sensing data, that are informed by the field data collection that we do. So basically, that is something that is being worked on right now and being incorporated into future versions of the regreening app that I talked about. So basically, not just using these tools as a way to collect data in terms of restoration activities that are happening on the ground, but also being able to provide feedback to the farmers, or it doesn't have to be farmers, it could be projects that are doing these interventions on the ground in terms of some of the important constraints in soil health wise within their intervention areas. Thanks.
Great, and I'll still for the same questions, a couple of new questions to you before I go to the ones on the chat. So there's a question from Emiru on how are these outputs related to human health? Is it possible to correlate and map with health indicators along the counties? And also, like a follow-up to what you just spoke on, how can we improve soil health in areas with high salinity? So maybe with the output first, and then the how to improve soil health.
Yeah, I think it's a great question. I'll ask Tor to tackle it first. I mean, this is what we're really trying to get at in this project, right? So how can we relate some of the soil health constraints or soil health indicators with human health, with crop productivity? So Tor, do you want to go first?
Yeah, sure, I can start. I mean, we know that, you know, like the health of the soil or the nutrients that are in the soil hugely impact also the health of the vegetation or the crops that we grow, and also by extension impact the quality of the food that we eat. So yes, it's very important to understand soil health and to sort of build healthy soil from the perspective of the nutritional security. And so I think that in terms of how the outputs relate, it's really around scaling these types of assessments to allow us to then combine this information with information on human health. So we have other groups within various projects, including Invisibilize, that work on different things that are not directly related to soil health per se. It could be nutrition, it could be human health, it could be mental health in the case of Visibilize. But having access to these maps and the indicators, the indicator maps, can be a very powerful way to look at whether there's any relationships in terms of human health and soil health at scale. So that's one of the kind of driving forces behind the development of these predictive models as well.
Great. And while I still have you to, or those also like a follow-up, like a second part of that question, like how possible is it then to map the indicators, the soil and land health indicators with human health indicators? So can they be overlaid across the counties that we just presented?
Yeah, they can be overlaid at even down to farm scale. So basically, the idea is that like the maps that we produce are at a spatial resolution that allows us to look at very localized variations in soil health, and that can be overlaid with other types of information, including on human health. And then, of course, at county level, we can aggregate these types of findings as well. So we can look at it at multiple levels.
Great, thanks. Lee, do you want to take the question on improving soil health? And then I see participant X has raised their hand. After I will invite them to actually unmute and raise their question in person. So Lee, over to you. And then we'll continue with the other questions.
Yeah, there's a lot.
Yeah.
I think the question about how we improve soil health with areas with high salinity is really interesting, because oftentimes, like if we think about Amboseli, that's a place with high salinity, high pH. So we're not going to be farming there. We're going to be allowing native vegetation to grow, like a particular grass species, Sporobolus vacatus, which is really tolerant. It actually only grows in salty places. So it's really important that we acknowledge and we use, for example, Tor's app called the Africa Tree Finder, where you can find which are the appropriate tree species that are indigenous to that area that can take the droughts. Perhaps we know that Balanites grows in high pH soils, volcanic soils. We know certain trees love these types of environments. So I think it's when we try to make high salinity drought-tolerant environments into something they're not supposed to be, that's when we have an issue. But one of the things that we can always do is compost. Compost is the best restoration technique. We can compost manure from goats, from cows, from chickens, with some, green material and some carbon source. And this is how we can really take soil stewardship to improve soil health. Thanks, Christine.
Great, thanks. I see participant X, and if you're able to unmute, please unmute and ask your question or your feedback. Give your feedback live. And there's also someone else. I'm trying to see who. So if you've raised your hand, please unmute and you can ask your question. If not, we'll continue with the questions that are already on the chat and the Q&A. Please, yeah, say who you are.
Yeah, my name is Jean Giro, and my question is to Tor. And you mentioned about an approach of testing soils that is faster than the conventional methods. Could you please talk about it?
Great, thanks, Jean. Should we take the other question and then we'll get to the answers? So there was a second person that was had raised their hand. Please unmute and ask your question, and then we'll take the two questions together.
Yeah, thank you so much, To and Lee, for the great presentation on LDSF. My question is really on this. Currently, as a project feasibilized for climate action, we are a bit interested on the 3 agroecological zones. That's arid, semi-arid, and sub-humid in Laikipia. And one of the problems we have been struggling for a long time is how to translate the data we have, the survey, like the findings we have, into something that the local people can use, the policymakers. And for this case, I wanted maybe if there is a way, Lito, from your hand, this data can really be segregated in terms of these agroecological zones, and the recommendation can be made available for the people who want to use them. And yeah, if the findings can also be kind of simplified for the local people on the ground to be able to kind of connect the dots and be in a position to kind of use the data appropriately. Yeah, thanks. But the other question I think I had posted on the chat is really on agroecological. It seems there is a lot on agroecology can borrow from the LDSF. So it means then if somebody has to do an agroecology activity, it needs first of all to have some data on soil health. It needs to know some pH of these areas where you need to do your agroecology and what have you. So my question then was, is this then means I need to do an LDSF first before really coming up with some interventions related to agroecology? Yeah, and thanks.
Great, thank you, Stephen. Over to you, Tor, and then Lee.
Yeah, sure. So on the soil analysis method, so what this is it's we use infrared lights to analyze the soil. So we basically scan this instead of using wet chemistry. We scan the samples that we collect using mid-infrared light, and that gives us kind of like a fingerprint for each soil samples and allows us to predict the different soil properties. And so the way it actually works is that we scan all the samples that we collect, and then we take a small set of samples, roughly 10%, and subject that to wet chemistry analysis to develop the calibration models. And we've been doing this now over a number of years and have a very large archive of these soil data at the lab in Nairobi. So basically we're now relying less and less on the wet chemistry, and that allows us to basically do more rapid soil analysis and also do it much cheaper than when we use the conventional methods. And also it doesn't involve any chemicals, which is really a positive thing.
Thanks, Tor. Lee, on agroecology and the intersection of agroecology and soil health, as well
Yeah,
as
so
on,
I wish I could show the sticker on the back of my computer about soil health essential to agroecology by Wazim. So I basically have said that you don't need to do an entire LDSF site. We can absolutely randomize plots in farms or whatever is appropriate. And yes, all these data can be used in an agroecology model. Your idea about simplifying these data for the farmers, I'd love to be able to do that here in this project. We can talk to Liz and work with you guys to do this, to bring this information in a simple format, preferably in Swahili or Ma or whatever is appropriate language. I think that would be absolutely fantastic. And thanks for the suggestion. Thanks, Christine.
Okay, and while I still have you, Lee, there was like a couple of questions on some of the graphs you presented. One was from Christopher on why Acacia seal and Passia americana are both in trees and species graphs, as well as a question from Charles on how was the dominant species cover assessed? Was it based on the number of individuals, frequency of occurrence, or the basal area?
Thank you so much, because I don't see those questions. So I'll start with the last one, because I think I already forgot the first one. You'll have to say it again. So basically, we have, do we have the plot graph? A plot is 1,000 meters squared from the center to the edge of the circle is 17.54 meters. So that's the pi r squared. So that equals 1,000 meters squared. So we basically set up four subplots in each of the plots, and we look at each of every single tree, every single shrub that's in there. So we count them. So it's essentially a count for each subplot across all the plots. And that's how we get to the frequency occurrence. So we're actually counting trees and shrubs and identifying them. I also thought that Charles's question on can observed species be linked to soil health? Absolutely.
Yes.
Absolutely. And this is a whole research area. What is the impact of tree species on soil health? What's the impact of soil health on tree species? And that's also why we're also promoting indigenous native trees that are suited to the area. I'm just going to keep going here. Jemima, if you go to our website that I put in the link, we post all of our sample costs for the lab, but it's $10.50 to do mid-infrared spectral testing. And that gives you about 17 variables that we predict from the spectrometer. You have to remind me, Christine.
Oh yeah, the other one was why the two species, so Acacia and Passia, are in both trees and shrubs.
It's just the height.
Yes.
So we're just trying to understand what's the shrub level canopy up to from 1.5 to 3 meters. And then if it's over 3 meters, then that would be more a tree species. So it's very possible that the same species could be this two different heights. That's why. Thanks, Christine.
Okay. Tora, there's a question for you from Evans Omondi, who asks, what machine learning model and soil data sets were used to make the predictions? And also, what is the specific spatial resolution of the final map?
Yeah, so in terms of the machine learning model, it can be a number of different ones, and it's actually something that the team works on all the time. So we use anything from very often it could be a model like a random forest model, it could be extreme gradient boosting, but also deep learning models. So it just depends on what models perform best for any given soil property or other indicator that we're predicting. So, but you know, if you are interested in like a general purpose model that works well in general, random forest would be an example of that. But again, it's something that's always evolving, and we're always improving the models that we use. The spatial resolution, the maps that I showed were at 30-meter resolution. The training that we use for the modeling is at 10-meter resolution, so we can produce maps down to 10-meter resolution. But in most cases, we produce them at 30-meter resolution for these types of projects. But it depends on the application again.
Great. Thanks, Dor. And these are the last two questions that are related to what you talked about on infrared, and it is how applicable is the infrared light soil analysis method? Can it be used widely, for example, by county government? And related to that, can human health outcomes be inferred from soil health indicators? And yeah. And if so, which indicators are more strongly associated with health risks?
Yeah, so in terms of infrared light, yes. I mean, this is actually by now a very well-established method, and it can be widely used. Whether county governments should invest in their own labs for this is an open question. I think not necessarily, because it's something, it's a method that allows us to do very high throughput analysis. So it means that you can, for example, within Kenya, you could have fewer labs that are doing this because you basically have such high throughput. At ICRAF alone, we can analyze between 35 and 40,000 samples a year. So there is really no need for all the counties to have their own labs. That said, we do support labs, and Lee's team supports labs in a number of different countries, and not just in Africa, but also beyond. So yeah, so it's possible to set up. It's very much a methodology that is easier to replicate than the conventional soil analysis. Now, for the human health outcomes, I mean, this is very much one of the research questions in Visibilize. You know, this is something that we are now looking at in terms of the indicators that are more strongly associated with health risks. I mean, we know from literature some of these things, but this is definitely something that we're looking at as part of the project. I don't know if Lee wants to elaborate on that one.
Yeah, I just think that I'm really happy for the questions on soil spectroscopy. We now are moving away from wet chemistry. Soil spectroscopy is faster, more robust, efficient, and accurate. So it's really, Africa's leading the way in terms of soil spectroscopy. We kind of skipped the wet chemistry. So we're really proud to implement this. We would not be able to do an LDSF site if we analyzed all samples on wet chemistry. It would be $150 per sample versus 10. So we're reducing the cost by 90%. So this is really how we can scale soil health assessments. Over to you, Christine.
Great. Thank you so much, Lee and Tor, for the questions, and thank you everyone for the presentations. As you please continue putting your questions and comments and feedback into the chat. Sorry about that. But as we come to the end of the presentation, I'd like us to take a few minutes to reflect on what we've had. What are our key takeaways, right? How do we integrate these data sets like the soil? And some of the questions that are coming is, how do we integrate these data sets across the project outcomes? So going back to the same Menti that we had before, so you can scan the QR code or go to menti.com and put in the code below. We'd like to hear about what are your key takeaways from the presentation, from the discussion, from the results that you had, but also then how do we integrate these data sets across the project? So let's take a minute or two to just answer the two questions on Menti, and then we'll wrap up. But also, if you're having a challenge accessing the Menti, feel free to type in the responses to the questions in the chat as well. So one key takeaway is, well, health is key in food production. I don't see any other. A few more seconds to just, note down your key takeaway. What is the one thing that will stay with you that you're taking away from the presentations, from the data, from the discussion? I don't see in the in the chat. Is it possible for, yeah. All right. I think, Lee, we can hand over back to Felicitas to, tell us the next steps. But thank you from myself for your participation, for the wonderful questions, but also, for the discussion. Lee, Tor?
Yep, thank you so much. It's been a real pleasure. And I think we're also pushing the boundaries here. We're really thinking about, are there soil health indicators that can be proxies for human health? How can we assess the impact of climate on soil health? How can we use these data for agroecological suitability modeling? I think this is all fantastic. Like I said, please feel free to email me. Christine, Tor, and I, we can welcome you to the ICRAF campus. You can tour the labs. And yeah, I think it would be really great to extend this collaboration. And I really thank Felicitas and Liz for encouraging us to do this webinar. Thank you so much for your leadership. Thank you. Over to you. Tor, maybe you say something and then over to Felicitas.
No, I just want to say thank you and great session. I look forward to future engagement. Thank you.
Yeah, thank you so much. And thank you, Christine, for heavily facilitating the session of the presentations. That was quite good. So thank you so much. And thank you, Lee, and for the participants for the active participation. Dr. Liz, do you have anything to add? And then we'll have our project manager, that's Dr. Alice Karanja, who will give us the closing remarks. Thank you.
Thank you so much, Lee, Christine, and Tor. It was really nice listening to your presentation and discussion about soil health. And I hope everyone has benefited from this, and let's continue engaging. And I really like you have a lot of resources on your website, and including this regeneration app, which people can engage with. So hopefully people will be able to engage with as we move forward. So thank you so much, and thank you so much, Feli, for spearheading this. Thank you.
Thank you, Liz. Over to Alice, who's our project manager.
Wow, okay, great. Thank you so much, Felicitas. Do I really have much to add? But really, as we conclude this important webinar on advancement in soil and land health monitoring, allow me to really express and join my colleagues in expressing our sincere gratitude to our presenters, Drs. Elizabeth Kimani, Lee, Tor, and Christine, for the depth of insight shared, and to all of you who joined and engaged in this discussion. Personally, I had been looking forward to this session, and I leave here feeling deeply informed and inspired, and I believe you all are. Today, we have been reminded that healthy soils are the living foundation of our food systems, biodiversity, and climate resilience. And we've heard how tools such as the LDSF and citizen science really are in transforming how we measure and manage land health. And I believe that the real frontier now is not only advancing the science, but ensuring that this evidence is translated in policy, practice, and also for community action. So when you think about the Eastern African drylands, this is not abstract. Land degradation, climate change are already driving food and nutrition insecurity, as well as mental health challenges in these communities. So the project is working to make these linkages visible, as Dr. Liz introduced, demonstrating that ecosystem health and human health are inseparable. And also, as it was highlighted in the beginning of this webinar, is that this engagement is a continuous process. We have ongoing public and stakeholder activities for engagement at the county level, the national level, and also the regional levels, as we are also in Eastern African countries, including Uganda, Tanzania, and Ethiopia, alongside primary data collection on nutrition and mental health in the Kenyan drylands. And these engagement efforts and data sets will be triangulated with, for example, the LDSF data and the remote sensing technologies that Lee and Tor and Christine have presented. And as someone asked, I think it was Professor Emiru, can be overlaid with the health indicators to further strengthen an evidence base and also the dialogues. So please also be on the lookout for our upcoming webinars and a soon to be launched project website, which will serve as a hub for visibility, communication resources, and ongoing updates from the consortium. So to conclude, on behalf of APHRC, CIFOR-ICRAF, and the Visibilize for Climate Action Consortium, I really thank you all for being part of this dialogue. Let us live here committed to scaling soil health interventions and stewardship, and also strengthening the collaboration so that science truly serves both people and the planet. So I really thank you. Back to you, Felicitas, and many, many thanks really for your very smooth moderation. And also, Christine, really, you really are coming handy. Thank you so much. Back to you, Feli.
Thank you, Alice. You've said it all. Nothing more to add. We'll just share the video with the people who participated, and we've also been seeing some messages, guys, requesting if you can also share the PowerPoint presentations. So I'll get in touch with Lee and Christine and the CIFOR-ICRAF team to find out if it's possible, but we'll be uploading the video online and we'll be sharing the link with you. So thank you so much for participating and for your active participation and for staying with us until now. Thank you. Bye-bye.
Bye. Bye.
Thank you, Felicitas. Thank you, everyone.
Bye-bye.
Thank you.
Wonderful presentation and engagement. Bye-bye.
Bye.